Start the recording. No.
Great. So this will be a short introduction to image informatics. It will start very basic. So I'm sure most of you already know a lot about it, but I just want to make sure that we're all on the same page for the rest of the course. Because images will be used quite a lot in spatial omics data. First, they can be like the input data, for example, for in situ sequencing or most of the method that Lars just talked about in the previous course. But we also use it for like doing a lot of analysis. Well, first for decoding the data for the methods that were just mentioned, or if you want to do segmentation of the cells, or if you want to look at the morphology of the tissue and so on. We also use them for quality control once we did that, or just to display the data on top of the image and check that it makes sense. And for simple visualization, for example, in spatial transcriptomics, you have the DAPI, the H&E image that you want to see under the markers and your data that you analyzed. So all this means images are at each step of the course we will present here. So I will start with the very, very basics of what is an image, right? So first, when you want to image something if you have a microscope, if you have a scanner, if you have your tissue and you want to make it into a digital image, you need sensors to capture the image, so the signal. And then the output from each sensor is one positive, like fine value proportional to the received energy. So these are the pixels of your image. Your images are usually colors in RGB, so red, green, blue. So you have 3 channels, one for each color of your image. For example, here we have an original image, and then we separate it in the 3 channels, red, green, and blue. And each channel will have the amount of signal for this given color. The maximum is white and the minimum signal is black. So if you have like all 3 channels with the maximum, then you will get white value. And if you have the minimum, then you will get black. So how do you digitize an image? Well, you will need to transform your image, your analog image, into like things that the computer can understand. So you have 2 types of digitization. You will have the gray level quantization. So basically, you discretize the amplitude values, and then you have the spatial sampling, and that's how you divide into pixels. And you can have uniform sampling, so that's like the pixel are uniformly separated, or non-uniform sampling, but that we will not talk about here because that's quite specific. So here we will have like simple images with uniform sampling. So basically, at each spot, you do a sampling of the intensity of your spot, and then you also do quantization. So you transform the vertical lines here into with a quantization resolution, and then you get your digitization at the end. So here are some examples of like the spatial sampling. So the number of pixels you want. So here we have 512 pixels in width, and then we go down to 120, 30, 256, 120, 64, and 32. So you see that the lower we will get less details about your image. And on the other side, the gray level quantization, that will like make less grayscale colors visible. So here we have 256 different values of gray, down to 2 where it's only black and white. So you have to define your gray level quantization according to what you want to see or how the computer needs to measure the information. In microscopy, the images are often in 12 or 16 bit. That means that they're encoded on like 16 bits. That means you can have up to 65,536 different levels of gray. But what you can see visually is usually about 32 levels of gray. You can see a bit more if you just compare 2 levels of gray, but in general, like down to 32 levels, here, you still see almost the same thing as the original image. So here we have an example where we have 256 levels of gray, and then we lower one after one just to like 33, and we still see the image with quite a lot of details. And then when we go down to 9, 5, or 3, then we really start to see huge artifacts. So when you do image analysis, usually you want to keep the number of bits quite high so that the computer sees more like fine details between your input. But for visually, like just to visualize the data, then 256 values is usually way enough, and that's what computers can manage on your screen. So here are some examples of like the usual quantization levels we use. A binary image like a mask will have only 1 bit. So that's black and white. It's either inside or outside of the object you want to see. Then on 5 bits, that's what the human can resolve locally. 8-bit is what most images have, and actually what the screen can display. So that's between 0 and 255 values. Microscopes usually export images in 16 or like 32 bits, and that's from 0 to 65,535 intensity levels. And so that's a lot of like precision. And then when you have RGB image, then they're encoded in 3 times 8. So then that's 24 bits. And that's very, very common also. And then how do you decide for the sampling of your number of pixels? Then you have to think like, how will the image be used? What are the limitations? I mean, do you have limitations in memory or in the speed you want to have if you do image analysis, for example? And yes, do you want only like visual interpretation, or do you want the computer to do the interpretation and so on? Do you want to keep the color? Do you want to keep the spatial and gray level resolution? So all that are questions you need to ask when you want to convert your images into lower number of bits or lower number of pixels. And then I want to talk about compression also and image coding. So when you have an image, you have a lot of redundancy in the image. When you talk about redundancy, it's important to understand that information and data are 2 different things. The information that your image has is less than the amount of data you have, right? Because data is how you encode your information. But you can have way more data than the amount of information, because you can have redundancy in your data. You can have, and this redundant data doesn't give any additional information, right? It's only the same information again. So when you do image coding or when you do compression, then you try to reduce the amount of data that you have while you want to keep the information total. So you don't want to reduce your information, but when you want to reduce your data so that it takes less space in the disk or in memory. So then you try to reduce the amount of redundancy. And you have different types of redundancy. You have coding redundancy. So for example, some gray levels are more common than others. So if you have like more gray levels at some point than like white or black, then you can reduce this by reducing this redundancy. Or you have interpixel redundancy. So for example, if you have a whole area that is all black, then you don't need to encode each pixel separately. And then you have psychovisual redundancy, because as we said before, the eye can only resolve about 32 gray levels locally. So then you don't need to have this redundancy if you only want to visualize your data. And here we have some example of psychovisual redundancy. So if you only want to use the image for visual observation, then you can reduce the amount of data a lot and still have something that looks pretty similar. For example, here we have an image that is uncompressed on the left, and that is 50 kilobytes. And on the right, we have the same image compressed with JPEG. And then we can a bit see some artifacts, but it's still a lot of the information is preserved for just visualizing your image. This is what we call lossy compression. So I will now describe the two kind of compression we have. We have you have either reversible or lossless compression. That means you didn't lose any information when you reduced the amount of data. It's exactly the same image. And you can then go back to the original, like you can do multiple lossless compression. It will always be the same image, right? You can win like 2 to 10 times when you do lossless compression compared to the raw data. And then you have lossy compression or non-reversible compression. And that's what you use when you want to put image online, for example, because you don't really care that it's the original image, or when you do videos or things like that, when it's only for visualizing the image. And then you have a compression ratio that can go up to 10 or 30 times the original data, depending on how much you can make it less nice. So here are some files formats that you see when you do lossy compression. Usually the most used is JPEG for joint photographic expert groups. And here you have really some artifacts that are visible when you put high level of compression in JPEG. So JPEG, you should never use it when you want to analyze your images automatically with the computer. You also have JPEG 2000 that was created a bit after JPEG based on wavelet transform. And then you have, but it didn't work that well. So you don't see that many images, and it's not used, for example, on the web. It's difficult to visualize the JPEG 2000 files. But if you ever find JP2 or JPX extension, then that means it's encoded with JPEG 2000. And then we have the lossless compression that we will use way more often during this course. And that are like the TIFF image, GIF or GIF, depends how you want to say it, or PNG. Most of the files we will use here are TIFF, and you have different kind of TIFF. And now you also have the OME-TIFF, where you can have annotations with it. But basically TIFF is very flexible format, where you can have 8-bit, 16-bit, 32 bits. You can have many channels. You can even have multiple pages and different levels of compression. And here you can also define if you want to have lossless or lossy compression. So you can have basically any kind of data inside of your TIFF. So when we'll have data from scanners or after your acquisition, we like to convert it to TIFF so that we can load them in any of our softwares or any of the image analysis tools we will use. So you will find a lot of TIFF in this course. GIF also supports animations, so it's nice for like on the web if you want to show something with a bit of animations. And then you have PNG that is also have the advantage of dealing with transparency. So for the web, it's very practical to be able to remove some part of the image and just show the background instead. And then you have another kind of format that we will not use this week, but it's interesting to know. It's the vector-based file formats, which means here, instead of having each pixel defined in your image, you will define the shape of your object with like lines and curves and so on. And then this has the perfect resolution at any scale because it doesn't like define individual pixels. For example, you have PostScript as a vector-based file format, or EPS, or PDF, or SVG. And I will not go into details, but yes, the one that is mostly used today is SVG when it's only image, PDF, of course, when you have other kind included inside. So yeah, just so you know all types of image that exist. And to show a difference between lossy, lossless, and vectorized image, here you have the same image in the 3 types with some zoom. And you see with the lossy that you can you can see the artifacts of JPEG compression around your image. So if we had to do some image analysis here, the computer would find these artifacts and would maybe like make false positives or problems around these. So that's why we always want to use lossless compression when we use it as input for our algorithms. And then the SVG, of course, has no pixel visible at all because it's only the shape that is defined. Okay, so how do you choose the image file format? Well, here we want to do some analysis of our images, so we will use lossless formats. But if you want just to visualize the image, for example, for spatial transcriptomics, when we display H&E under our data, then it's fine to compress it with, for example, JPEG. If you want to have illustrations or other, then you will go for lossless formats such as PNG or vector format. And then I can mention that when you want to open your images like TIFF or OMITIF that I mentioned before, one very good library is called Bioinformatics. And it's a software that you can use for reading and writing your images in open formats. It can also read many proprietary formats like that we will find during the course. So if you install Bioinformatics, then you can use it in many tools like ImageJ, Cell Profiler, IC, Omero, and so on, or MATLAB that you will usually find when you do image analysis or when you do decoding of data like we will do this week. You can visit the web page here and see how many formats they can actually read and export to. So that's one thing. And then the formats that we will see during this course, of course, there will be many image formats. So I talked about TIFF, OMITIF, JPEG, but also like proprietary formats from companies that do the scanners or the how you acquire your image. For example, a lot of them here will be in CZI, which is the format from Zeiss, and that contains images. But it's very easy to convert the CZI images into TIFF, for example, using the the Bioinformatics library. Other kind of formats that we will find are not directly images, but can contain images. For example, we have annotation formats, and we will find some at the SpaceTX format, which is a format used by Starfish for in situ sequencing data or other kind of image-based spatial omics. And we will see today during the labs how to convert formats into SpaceTX or from SpaceTX into open formats or like other formats that we can read and write. Then you have other annotation formats like Surat or Andata. Surat is used in R and Andata in Python. So this week we'll focus more on Andata because I don't think we have R labs. And in the Andata format, you have a lot of annotated data. It looks like this, just to have a fast overview, and we will go more into details during the labs and during the course on that. But basically, you have observations and variables inside a huge matrix here, and then you have some annotation for each variable and for each observation, and you have unstructured annotations. The images, for example, if you have spatial annotations, then you want to display the image under, then the image will be in the unstructured annotation. And we will see how to create these Andata and how to display them, how to visualize them in simple ways. So any question on that part so far? Or comments or things that are not clear? Else I will move to the next part, which is how to visualize all these objects and images. Okay, So yes, now I will focus a bit more on the quality control and visualization of our images and of our annotations that we had. And for that, I wanted to introduce a tool that we developed internally in collaboration with the Velby lab in a project that is called Tissue Maps. And this tool is for interactive visualization and quality assessment of large-scale spatial omics. It has been, there is a bioRxiv paper on the tool if you want to read it, but I will show you today how to use it, how to install it, and what you can do with it to display images, but also different kind of data. It's an interactive viewer, which means you can install it locally. I mean, you have a desktop viewer and a web-based viewer. So you can just go to a web page and visualize the data with tissue maps, or you can install the software and load your data on your computer. You also have Jupyter Notebook integration, and that's what we will use this week. So it's already installed as part of the environment you installed in the pre-course. And then you can visualize your images and data directly in the notebook and interact with it. You have different ways of installing it. In the environment, we use pip, but if you have Windows, you can also install it directly with the Windows installer, and then you will get the graphical user interface. You also have a Docker image if you want. All of that is available on the web page, and I will give you the link at the end. So the challenge with tissue maps is how to view and share large spatial omics data sets without having to download the huge images or the markers and everything. And so that you can interact with the data. You can also do spatial statistics, define region of interest, play with clustering alternatives, and so on and so on. Here is an example of how you can like interact with the data, zoom in, zoom out. You have like subcellular resolution for in situ sequencing data, or display the different cell types with spatial statistics. So all that you get when you use the tissue maps viewer. Tissue maps can load 3 types of data, mainly images, and we talked about images before, but tissue maps can load 8-bit images for now. We are working on making it possible to display a higher range of data, but like the best is to convert your data into 8-bit before you load them. We can also display and draw and import regions in tissue maps and markers, and we can display millions of markers on top of the images, so we are not limited to the number of markers or number of categories of markers. And we're also working, and I will show you some examples on how to load directly data objects where you have your images and markers, annotations in the annotations of the data. So just to show you very fast, if you have 2D images, you can like have multiple layers of images also and display them and filter them based on some simple filtering and add colors to them and so on. So that's very practical if you want to check, for example, if you have registration between images, you can display them on top of each other and then see that they are well aligned. We also can load markers. So if you have gene expressions or cell types or any kind of data, you can load them from CSV files where you have X and Y coordinates, and you can have like a gene types, and then it will like put one color and one shade for each marker type. You can also have a bit more complex information in the markers. For example, you can load pie charts for each marker and then display the different the legend of the pie chart and how much you have in each sector of your chart, and that for millions of markers again. And finally, you can have regions. So you can draw regions on top and analyze your region so that you can extract how many of each gene you have in your region, or you can like import the regions. So here, for example, we draw 2 regions and then we can analyze them and see how much information we have in each region and do some more advanced analysis on these things. We can also import regions if you have them in GeoJSON format, for example, from QPath or CellPose or etc. They can export the regions directly, and then you can display them and again analyze what you have in each region. We try to make tissue maps compatible with as many open source tools as possible, so that you can import and export from these tools and display in tissue maps and do your analysis or visualization online or locally. So tissue maps is as modular as possible and then can connect to all of these different tools. And now I wanted to make a bit of a demo of what we can do with tissue maps. But if you have questions also, don't hesitate to interrupt and ask. So first, I want to show how you can use tissue maps when it's online as a tool for like sharing your data with others. And for that, we can look at the Human Developmental Cell Atlas, where we displayed some lung data set at different weeks of development. And I will not go into details, but you can check different kind of analysis. You can look at UMAP data from single cells, or spatial transcriptomics data, or in situ sequencing data. And all of them have like different ways of being presented. So for example, here, if we look at single cell data, then you will have a view of the UMAP cell types. And then you can see, like, for example, if you want to see the MESCL type, you can look at where it is, or you can like put it on and off and play with your data. You can also draw regions and export them and so on. So you also have spatial transcriptomics data available, and then here we can either display the cell types for each spot of the spatial transcriptomics that was analyzed, and then you can look at the different probability of cell types for each of the spatial transcriptomics spots, or you can display gene expression directly from the raw data, and then you get like for each point the value of this gene for this spot. In the same way, you have in situ sequencing data. Oops, sorry, here. Where is it? So we can look at PCW6, for example, and that's the data we will play with during the lab today. So it's 6 weeks of an in situ sequencing data. And then if we display the raw reads, then we will get like the mRNA points. And then you can see how they are with subcellular information. And then for each, you have the targeted name of the gene. And you can also see where they are in the whole tissue. Then you can like see the cell types after segmentation. And then you get like one pie chart for each cell segmented and display it on top of your tissue. So here you see how the cell type are the same as in the spatial transcriptomics. Same for screenshot data. So this was just a fast overview of how you can present your data online. But then we can look at when you're trying to make this kind of presentation, then you want to load your data locally and then create a project and then push it online. So then we can install the tissue maps viewer, and you see you get a nice graphical interface here where you have different options, and you can load your data. So for example, here I have a TIFF file with DAPI layer, and I can directly drag and drop the TIFF file into tissue maps, and then I can like zoom in and see the raw data. And then let's say I want to display some genes on top of that. So I have a CSV file here, and we can look at what it contains. So if I open this, maybe I can open it with Excel here. And then we can see what we have in this tab. So we have only 3 columns, one with the gene names, one with the X coordinate and Y coordinates. So let's just do the same, drag and drop that here. and then I can select, so my X coordinate is in column X and Y in column Y. And then I want to group values by genes so that we have different colors for each gene in the data. And then I load it. And then you can, very similar to what we had on the browser, you can see like the points on top of the data, and you can like see for each point where it lies. So then you created a very simple tissue maps project, and you can save it into a tmap file, so that when you have a tmap file, if I open this tmap file here, I will already have my cell types that will load. And this one actually I created with pie charts instead of cell types, so I can look at the different pie charts. But it's a similar way. You just create your project, and then you get a tissue map file. And then you can actually export this into a static web page so that you can share it online to others. Everything is web-based, so everything that is available in the application can be shared with anybody else online. You can also make screenshots. So if you like, let's say you want to have a nice view of this thing here, you can capture the viewport at a high resolution, and then it will like take snapshots of all your data at a higher resolution and make an image out of it. So for example, if you want to publish it, you can have a very, very nice figure directly from your data in your paper. And yes, and then it will save it as an image, and then it will open it, and then you can, yes, and then you can share that, and you see that it has quite high resolution image, so it's very nice for publishing or for putting online for illustration. Good. Then I can continue by saying that Tissue Maps is also available as a Jupyter Notebook extension. Oh, wait, just before that, I said earlier that we also could load and data objects. So I can show you some example of that. So let's say we have H5AD objects, which are like directly and data files with annotations. Then you can actually drag and drop these into tissue maps and have a look at what it contains. So for example, I downloaded the mouse brain data from a website. And then you can see it doesn't have images here, so it doesn't load anything, but you have many observations that you can display. For example, the number of genes by count on the data, or the total counts of genes, and so on. Or you can also look at annotations if you have annotations. So here we have like different values and so on. So then you don't have to set up your project with anything else. Just you click on one of the objects and it will display it. So here you can also look at the gene expression. So you will have it on top of the data. It can also contain multiple images. And for example, here I have one data that I created myself, where I have like, I think 9 different images. It takes a bit of time to load with Zoom, but yes, so here I have my 9 images. And then the same way you can like display your annotations from the Python objects directly on top of the data and have different clusters, or look at the quality control, like number of counts on the data and so on. And then you can like see how many counts you have for each point, or you can display the raw gene expression on top of the data. And then in the same way, you can then export to a static web page and share it with collaborators. Or for publication, if you want to publish your results, you can publish your data objects directly in the browser and share it. You can also look at it in tissue maps in Jupyter Notebooks. And then I will do a small demo of what you can do in the notebook. So And we will see that later today in the labs also, where we will use tissue maps for visualizing the data, for quality control, and for some like plugins also. So first you import the tissue maps viewer, and then it will tell you which version you're using. And then you can define, for example, here I have an image and the CSV file, and I want to load them. So then I will call load data in tissue maps with my images and CSV files. And then I will say that my selector for the X column and Y column is called X and Y. And I want to group by the column cell type. I want a scale of 1.5, and I want to use these plugins inside. So tissue maps, I will talk about plugins a bit also, but tissue maps can load some plugins that you can develop or take directly. And then you have your tissue maps viewer directly inside of the notebook, and it's interactive. So you can zoom in, you can like click on one object, and then you get like the cell types for this one, or you can visualize things. And you also can run the plugin. So for example, here, if I run the feature space plugin, I have an error because I didn't load it, but I will show you that outside of the Jupyter Notebook, I don't have this plugin installed here. Any questions so far? Or comments? Okay, then I will continue and talk about the different plugins that you can use and install in tissue maps. And I tried to run the feature space plugin here, but I forgot to install it. So I will just show you how it works. You also have a 3D version of it. Points to regions if you want to convert your, for example, in situ sequencing data to different regions automatically. Yeah, spot inspector I will talk about also. So the feature space is if you have on top of your data, you also have like UMAP coordinates, then you can display next to the data the UMAP coordinates of the same spots. And then you can interact. We try to make everything as interactive as possible. So you can select points in the left side and see where they are in UMAP, or select points in UMAP and see where they are in the tissue. You can also change the colors or anything, and it will change on both sides. So this is like interactive feature space visualization of your data, and makes it very easy to understand where your data is and what kind of cell types you have, for example, here. You also can display it in 3D if you want, so you can like make nice videos if you want to like share your UMAP feature space, or any other feature space actually. This is only for like displaying. You cannot select things in 3D. So if you want to really do interactions, then it's better to have a 2D version of it, because it's always simpler to select things when they are in 2D. And then we have a small plugin that was developed by Axel Andersson here at the lab, where you take in situ sequencing data, for example, as input, where you have embedded vectors for each spot, and then you can create, or like targets from each point, and then you can create automatically a number of regions that will cluster your points depending on like local information. That's an example, of course, but you could think of any other kind of plugins that you could develop or contribute to. And then the last one is one that we will actually test during the lab today. It's when you do in situ sequencing decoding, then you want to know if the spots you find are actually good in the raw data, so in the original images, by looking at the intensity of the pixels in each round and channel. So for example, here we have 5 rounds and 4 channels, and you can see how the different points in the tissue were decoded. So if you click on this one, you will see that it will follow the spots in each round following a code book, and then you can decide if it's a bug or if it makes sense, and so on. So this is very practical for quality control when you do decoding of data from multiple rounds and channels, because it's very difficult to see. Here we have like 20 different images on the same point, and it's difficult to see the 20 images at once when you have that amount of data when it's on the whole raw data. So that we will do actually during the lab today or tomorrow, depending on how fast we go in the lab. So just to conclude on tissue maps, it's really a web-based viewer. So it's meant to be used in the browser, but it's also possible to install locally. And it is for interactive viewing of large scale 2D images with regions and markers on top of it. It's a collaboration with other open source initiatives for cell and tissue segmentation and spatial statistics. And we try to make it as compatible as possible with other tools. We don't want to like reinvent everything. We just want to see tissue maps as the viewer for spatial omics data, and then you do all the analysis and everything else in other tools and display them in tissue maps. And we are open for like ideas or comments or anything, so you can actually contact us. Or if you find bugs or if you have feature requests, you can also use a GitHub page or the image.sc forum, where we have a section for tissue maps.